All right. So I am Alvar Akil from the Gokchaman Lab at the University at Buffalo. And the title of my presentation is Balancing Selection on Genomic Deletion Polymorphisms in Humans. So the first thing that we ought to discuss is what balancing selection is. So let's suppose an allele emerges in a population. One of many things could happen to it. Let's say the allele is disadvantageous or deleterious and is subject to negative selection. It is going to quickly be eliminated from the population. If the allele is advantageous and subject to positive selection, as shown in blue, the allele will quickly rise in frequency all the way to fixation. However, even in the absence of these selective constraints, an allele is expected to fix one way or the other, elimination or fixation, given enough time, just by drift. However, sometimes it is possible for an allele to be maintained at an intermediate frequency for an extended period of time in the face of drift. And the force that is responsible for doing that is called balancing selection. So in general, the older a polymorphism is, the more likely it is to have evolved under balancing selection. And scientists in the past have used the same idea to study balancing selection in humans. And in particular, they focused on polymorphisms that emerged prior to 5 million years ago, which is our divergence time from the chimpanzees. So they reasoned thus. They said, all right, there are a bunch of genomic polymorphisms in humans. And so if the same polymorphisms exist in chimpanzees as well, then by parsimony, the polymorphisms must have emerged before the divergence time of 5 million years ago, and therefore they would be ancient and therefore more likely to be under balancing selection. Now, this is a great approach, very few false positives, but it does have a false negative problem. So we used a similar idea, but we leveraged some of the archaic hominin genomes, high coverage archaic hominin genomes that have become available over the years. So here's what we did. We focused on the aggregate of human genomic polymorphisms, and we checked whether the same polymorphisms were also present in Denisovans and Neanderthals. And if they were, then barring introgression and recurrence, we concluded that the ancestral population of the two lineages, which existed sometime before 700,000 years ago, should also have the same polymorphisms. However, if the same polymorphisms are not also present in the chimpanzees, then we can reason that the polymorphism emerged at some time during 5 million years ago, our divergence from chimpanzees, and 700,000 years ago, our divergence from the archaic hominins. So now the question that we must ask ourselves is whether this time depth is good enough to cause an overrepresentation of balancing selection candidates. And one way to test for this would be to first calculate the proportion of all human polymorphisms that are ancient, older than 700,000 years ago, and then compare this empirical percentage against the percentage expected under some sort of neutral condition. And that is exactly what we did. So on the x-axis of this graph, we have the percentage of human polymorphisms that are ancient, older than 700,000 years ago. And along the y-axis, we have results from different simulations. And the simulations are guided by three main parameters: the effective population size of modern humans, the effective population size of archaic hominins, and the time of divergence between these two lineages. Now, the vertical line you see over here represents the empirical percentage of human polymorphisms that are ancient. And what we see from this graph is that under most simulation models, there is an empirical excess of ancient polymorphisms. The only cases in which this does not hold is when we tweak at least two parameters in very unreasonable directions. So what this graph really tells us is that this time depth between 5 million and 700,000 years ago is good enough to study balancing selection. Now, instead of focusing on single nucleotide polymorphisms, which are usually studied in selection studies, we focus on somewhat underrepresented part of the genomic tapestry, the structural variations. And in particular, we focus on deletion polymorphisms. Now, the problem with studying deletion polymorphisms in this context is that we have the deletion data for modern humans, but we don't have this data for archaic hominins. So we needed to find a way to identify deletions in the archaic hominin genomes, and we do this using a read depth-based approach. So over here, we have 4 panels, and each panel corresponds to one archaic hominin genome. There are 4 high-coverage archaic hominin genomes, and we're looking at a window of genome that is polymorphically deleted in modern humans. And so in 2 of these cases, we see that in 2 of these archaic hominins, we see that no reads are mapping to the window of deletion. So this is very likely a homozygous deletion in these archaic hominins. In one case, we're seeing that the read depth, that the amount of reads mapping to the window of deletion is more or less the same as that's mapping to the region around the window of deletion. So in this case, a deletion very likely does not exist. And then there's an intermediate case where the read depth is sort of an intermediate level between the 2 cases previously described. So this is very likely a heterozygous deletion. And so we bioinformatically automated this process of identifying deletions and managed to identify a lot of deletions in archaic hominins. And using this method, we reached the following result. We found that out of all the deletion polymorphisms we looked at in modern humans, about 575 of them are shared with archaic hominins. But only about 430 of these 575 shared deletions are truly ancient. The rest are a result of introgression from archaic hominins into moderns or independent emergence of these deletions in both lineages. So now that we have identified these ancient deletions, we ask the following question. We ask whether these ancient deletion polymorphisms are more likely to be targets of balancing selection than more recent deletion polymorphisms. And so the way we use to test this is the following. We introduce a concept called Goldilocks drift, and we're doing this to identify signatures of balancing selection. So let's assume that a deletion emerges in a population and it's subjected to balancing selection by nature. And let's also assume that this deletion rises up in frequency until it reaches a certain equilibrium frequency. Now that 2 haplotype classes have been established, one with the deletion and one without, SNPs, single nucleotide polymorphisms, can emerge on the haplotypes. And some of these SNPs will drift up in frequency, but the maximum frequency to which they can drift up is the same as the equilibrium frequency of the deletion. So under intense balancing selection, what we should expect to see is 2 haplotype classes such that the variation within each haplotype class is minimal. And so it turns out that there is already a way out there in previously published studies to measure the intensity of the so-called Goldilocks drift. And that statistic is called beta. And so over here, there are 3 graphs, 3 panels. The top panel is an African population, the middle panel is a European population, and the bottom panel is an East Asian population. Along the x-axis, we have got the beta statistic, and along the y-axis, we have got labels as to whether the deletions are recent human-specific deletions or ancient deletions shared with archaic hominins. And across populations, the trend that we see is that ancient deletions have higher beta values on average, meaning that they're more likely to have evolved under balancing selection. So the last question that we ask is whether these ancient balanced deletions are more likely to be associated with certain phenotypic categories than more recent deletions are. And it turns out that they are. In this graph, along the x-axis, we have different phenotypic categories, And along the y-axis, we have a certain enrichment ratio, asking whether ancient deletions are enriched for certain phenotypic categories compared to non-ancient deletions. Dark orange color over here represents that the results are significant, and light orange represents that they're not statistically significant. And so what we observe is that these ancient balanced deletions are phenotypically enriched for categories involving immunity and metabolism. So many of the deletion polymorphisms associated with immune and metabolic disorders have in fact evolved under balancing selection. So with this, the takeaway message that I want to give is that, number one, many of the deletions that are associated with the various metabolic and immune disorders are very old. In fact, Neanderthals and Denisovans had those deletions as well. And secondly, that these old balanced deletion polymorphisms are very much part of what makes us human. Usually, when we talk about variants that make us human, we're talking about variants that are fixed in humans to the exclusion of other groups. But it turns out that if certain polymorphisms are present across human populations as well as Neanderthals, then they may as well be considered part of what makes us human. If anybody is interested in learning more about this study or wanting to know about more technical details, the paper is out on eLife, and it'll be great if you could give it a read. That's all.
Excellent talk. Next, we have questions. So if you would like to be recognized to ask a question, please use the raise hand function, and we will ask you to unmute so that you can ask that question. If you're feeling shy, you can put your question into the chat box where we'll read it off so it gets into the recording of this session. I guess everyone's probably still feeling a little shy. I guess I'll get us started with a question. So this is obviously very, very cool stuff. What are you going to do with it next? What's the next direction?
I think with regards to balancing selection, it's pretty much done. So I'm moving on to other things for now. But if there is any sort of criticism or critique, then I'd be happy to kind of work on it further.
Yeah, do you intend to stay in humans or?
I usually tend to stay with human evolution, but I'm a bit all over the place. A bit of ancient DNA, a bit of PubGen, a bit of cancer genomics, and so on.
That's exciting. So with, eLife recently changed their model. So is this published? Does that mean you could update if someone read your paper at this conference and had questions about it or anything you wanted to update, or what does that look like at this point?
I'm not really sure, because this paper was one of the last papers to be published under the old model. So I'm not sure whether it would apply to my paper too, but that's something I could look into.
Interesting. In the chat, yeah, would you like to read it out
yes.
on the video?
It says, are there any biases with the sequences obtained from ancient DNA that could influence your indels? There could be some biases. For example, if a certain region has a high percentage of repetitive sequences, then of course it would be difficult for reads to map onto that region. So it is possible that we might have called some deletions we might have called certain windows deletions in archaic hominins by error. So it is possible, yes. Thanks for the question. Anyone else?
I think we have to start our transition to the next speaker at this point. Thank you very much for taking that question.
All right, thank you.
Kees, would you like to put up your slideshow?
Yeah, is that all looking good?
That looks good, and you can start whenever you're ready.
Okay, wonderful. So I'm Kees. I'm a PhD student. We're at the end of my PhD now at the University of Bath in the UK, at the Milner Centre. And I'm going to be talking about some work I've done looking at the consequences of polygamy for molecular evolution in birds. So I'll start with just a quick kind of summary of what we mean by polygamy in birds, because there's lots of different types. So we're including in this polygyny, so that's when the males are mating with multiple females. And that includes kind of lekking systems, like you see with this bird of paradise, where the males all go on a branch and kind of show off and the females pick their favorite. But individual males can end up monopolizing mating opportunities through these systems. And it also includes systems like stable dominance hierarchies, like you see in the chicken. We also include in polygamy polyandry, so that's when females are mating with multiple males, like you see in the jacana. Often they kind of have a harem of males and they'll mate with each of them and produce a clutch of eggs with each of them. And the males will then do the incubation. And also kind of more niche strategies like double clutching polygynandry. So this one, a male and female kind of pair up, produce a nest together, and then both will kind of leave and try and explore additional mating opportunities before, I think usually the male eventually circles back to incubate the first nest in the end. So we're considering all these to be polygamy, and the key thing that they have in common is that relative to monogamy, all these systems are predicted to result in greater variance in reproductive success. And variance in reproductive success is quite important for molecular evolution. There's like a couple of different direct consequences of it. So one is that it lowers the effective population size. So you can see in this kind of example here, one male is monopolizing all the females. So these three males are not actually part of the breeding population. So the whole effective population size is lowered relative to monogamy if the four males were getting one female each. Also, greater variance in reproductive success results in greater sexual selection because the potential fitness benefits from mate attraction are much higher under a polygamous system than under monogamy. So this male is getting huge benefits from being the best male on the branch. Whereas in a monogamous system, that difference is not so extreme. And there's kind of direct consequences of both of these factors for molecular evolution. So we expect smaller populations to be less genetically diverse, less efficient at removing harmful mutations, also less efficient at fixing and spreading beneficial mutations, and less affected by GC bias gene conversion. So just very briefly, this is a meiotic repair bias, which pushes the genome towards higher GC content. The important thing about it is it's selectively neutral, but directional. And kind of all these different effects stem from the higher genetic drift in smaller populations. They're more affected by genetic drift. And that kind of random sampling of alleles in each generation is just removing SNPs at random. So it's removing more SNPs than it would otherwise. It's kind of inadvertently selecting for, or not selecting for, ending up with more common harmful mutations, inadvertently removing beneficial mutations, and also it's opposing these kind of any directional process it will oppose because it's just randomness being thrown in. Lots of theoretical work about these kind of links with small effective population size. In terms of comparative work in birds, it's mostly based on comparisons between the autosome and the Z chromosome. And what we see is that the Z chromosome, relative to the autosomes, is less genetically diverse, and it appears to be less efficient at removing harmful mutations. And this effect is more extreme in polygynous populations where the males are mating with multiple females. And in those populations, the male effective population size is particularly small, and the Z chromosome is generally found in males. That's where the evidence for this effect kind of is. And then thinking about the consequences of sexual selection, and this really depends on how sexual selection acts. So is it that successful males are fitter and healthier than the other males, or do they just happen to carry the right allele at some specific sex-selected loci? So alleles for long tails and bright coloration. And this is kind of the whole good genes theories of sexual selection versus runaway theories. And this has a big impact on the consequences for molecular evolution. So if, in line with good genes theories, successful males are generally healthier than other males, then populations with more intense sexual selection are expected to be more efficient at removing harmful mutations and also more efficient at fixing beneficial mutations. That's because this these kind of males which can monopolize all the females, there's a huge benefit to them being the healthiest individual there. So if this selection is for the healthiest individual, then they will spread those healthy genes much more efficiently than in a monogamous population where there's not such a big difference in reproductive success. Essentially, what this means is that the selection coefficient is just greater in magnitude in polygamous populations. There's lots of theoretical work with this as well, and lots of the kind of work outside theories come from experimental evolution studies. So where they kind of don't allow flies or ball mites to evolve under sexual selection, and those ones without sexual selection end up being less healthy as a population. Generally, it's very mixed, the results, but on the whole, that seems to be the trend. Okay. Oh yeah, and the other sort of sexual selection, so if the male is just carrying the right combination of a handful of sex-selected loci, we don't really have a clear prediction for molecular evolution. Those alleles will be under strong selection, but the genome as a whole, there's no real prediction. Okay, so quick recap, because I blasted through that theory. So more polygamy means greater variance in reproductive success. That means two things. One is lower effective population size, which means more genetic drift. And the other is it means greater sexual selection, which means more effective selection, essentially, more effective purifying positive selection if natural selection and sexual selection are aligning and selecting for the same thing. Okay, so now onto the study. So what we did was look at 150 individual bird genomes, largely from the BTNK project who are collaborating on this. It includes 121 monogamous and 29 polygamous species. And we define polygamy in a way that it includes all the different types of polygamy I talked about at the start, by defining it according to the most polygamous sex. And then we analyzed the SNPs to extract three measures. So one was a measure of purifying selection effectiveness. That's just the ratio of non-synonymous polymorphisms to synonymous polymorphisms across all the autosomal genes. One was the measure, so we expect with better purifying selection, more efficient purifying selection, the non-synonymous polymorphisms are selected against because they're generally harmful when they first pop up. And we also had a measure of neutral genetic diversity, that was just intergenic heterozygosity, quite a standard measure. And we also had quite an unusual measure of GC-bias gene conversion effectiveness because of the data set we had with long branch lengths. We didn't use a lot of the normal measures. Essentially, this is the ratio of SNPs affected by GC-bias gene conversion to SNPs not affected by GC-bias gene conversion. And because most SNPs that are affected by GC-bias gene conversion are selected, kind of fished against by this process, hesitant to say selected, but sort of selected against the GC-bias gene conversion. A lower value of this ratio statistic means that GC-bias gene conversion is more effective, just like a lower PNPS means more efficient purifying selection. Okay, so on to the results. So first of all, we do see that polygamous species carry significantly fewer harmful polymorphisms relative to neutral polymorphisms than monogamous bird species. So you can see here, polygamous species on the right, monogamous on the left. Each dot is just a species, very straightforward. The p-value comes from a PGLS model, so phylogenetically controls, and included covariates of body mass and geographic range size to control for effective population size variation, which we weren't interested in unless it was caused by polygamy. Okay. So this is not consistent with lower effective population sizes in polygamous species, but is consistent with the alignment of natural and sexual selection and more efficient selection in these species. The second result is that they have pretty similar neutral genetic diversity to monogamous species, same sort of model, no significant difference here, although a trend towards them being more heterozygous, the polygamous species. And similarly, no significant difference in the effectiveness of GC-bias gene conversion, but again, we see a trend towards more efficient GC-bias gene conversion in the polygamous species. So these non-significant trends had us slightly worried because this is a comparative study using available genomes. It's very possible that we'd inadvertently sequenced polygamous species which had particularly high effective population sizes compared to monogamous species. And this is kind of the pattern of results you'd expect to see in that case, except you wouldn't necessarily expect this one to be stronger, but maybe this is just a less noisy measure, the PNPS ratio. So we wanted to look into this a little more closely to ensure that wasn't the case, because we're really interested in the effect of sexual selection. And we basically found that it's unlikely to be that. So this is kind of a graph where the dots, the larger dots are larger effect sizes and the darker dots are more statistically significant effects. And you can see that the covariates of body mass and geographic range size. So smaller body mass is consistently associated with larger effective population size, so smaller birds of larger populations. And we see this effect coming through all our different measures. So they have more efficient purifying selection, they have greater genetic diversity, they're more efficient GC bias gene conversion. And similarly, we see this very consistent effect of geographic range size, where birds with greater geographic ranges have more efficient purifying selection, greater genetic diversity, more efficient GC bias gene conversion. In contrast, the effect of polygamy is very specific to the effect of purifying selection. So that is very consistent with an effect of sexual selection, which wouldn't be expected to affect genetic diversity or GC bias gene conversion. And another thing to note here is that the effect of polygamy is on a similar scale to that of body mass. It's kind of very well recognized that smaller birds with large populations are more efficient at removing bad mutations. We're seeing a similar effect size here of polygamy. Okay, so on to the conclusions. So polygamous species exhibit stronger purifying selection than monogamous species. This effect is unrelated to the effective population size and instead reflects the alignment of natural and sexual selection and the purging of harmful alleles from the population. So in these polygamous populations, because only the best individuals are breeding of the more polygamous sex, it's purging out all those other kind of possible genomes which would be maintained in a monogamous system. We also find that the loss of effective population size associated with polygamy has very little effect on molecular evolution. And in combination, these two factors suggest that polygamous species may be better able to withstand the buildup of genetic load associated with long-term low effective population size by more efficiently purging these bad mutations as they pop up. Okay, many thanks for listening. Huge thanks to my co-authors on this work. This was published at the start of this year, so if you're interested, you can check it out. And huge thanks to my funders, of course. Thanks so much.
All right, wonderful talk. Looks like we already have a question in the chat, which I'll read out, so we have it on video. Are monogamous species more common in birds, or were there just more of them available for the study?
Yeah, monogamy is more common in birds in terms of how many individuals a male will partner up with or a female will partner up with. In terms of whether they actually only mate with one individual, promiscuity is very common in birds. We didn't really look at that because we don't have as much data on it. But yeah, biparental care and monogamy is common. How promiscuous they are is more up for debate, I hope.
Excellent. And if anyone has further questions, you can use the raise hand, at which point I will unmute you so you can verbally ask your question. If you would rather type it in the chat, you can go ahead and do that so that we can read that out. So it's in the video for our viewers at home at an unspecified later date. All right, well, great stuff. Thank you so much for sticking around to answer questions. So next up will be Daniel Stanton. So if you want to get your screen share up, I will let you know when you're good to start talking. And I realized I misspoke earlier when I said that we would have Berenice going at in the, well, I guess 4 o'clock. She will go in the last talk, or as the last talk of the session. So that's 2:45, not the top of the hour, as I had previously said. All right, Daniel, go right ahead.
All right. Hang on a second. I'll stop sharing real quick because my computer is telling me I have 3 different windows open, which is interesting. I apologize.
We always got to have a little
Okay.
surprise in the session.
I know, right? All right. So let's see. See a little bit of time. Let's try this again. Okay. I'm a little afraid to actually
Good.
share my screen and go to presenter view because that's where it screwed up. Can you guys see my screen? Okay, cool. That's where I screwed up the first time. All right, so today, like April said, my name's Daniel. Today I'm going to talk about how the Xenocoelomorpha, which is a really cool phylum, is a keystone phylum in understanding the molecular evolution of the circadian regulation. So let's start off by just talking a little bit about the circadian clock. So what circadian regulation in animals basically is a way for cells to tell time, to regulate different processes within particularly a light-dark cycle in most cases. They're influenced by different internal and external environmental cues, and they consist of a very kind of elegant core mechanism that is very complicated once you add in all the extra mechanisms that feed into it and how this mechanism feeds to other mechanisms as well. And so really what sets the tone or sets the timing of the clock is the protein dynamics of the negative regulators being made and accumulating in the cytoplasm and then being translocated into the nucleus where they can inhibit their positive regulators. So for deuterostomes, that's going to be clock and BMAL for positive regulators. For the negative regulators, you have PER and CRY. Most protostomes have clock and cycle. And PER and TIM act as the negative regulators, with CRY having a different role. We really don't know what's going on in the lower bilaterians. And from a little bit of research, we know that with cnidarians, we don't have a PER ortholog, but we have CRY orthologs for negative regulators, and we have a CRY in sponges. And we also have some positive regulators in those particular species. So let's take a big picture look at the evolution of the circadian clock negative regulators. And so I'm not going to go through every single species in here, but I want to kind of point out some keys. So we have the deuterostomes, protostomes, cnidarians, and then everything else. So really what we're focusing on is the center of all this. You know, what happens at the base of the bilaterians? And so if you notice, like I said earlier, you know, clock or BMAL or cycle, we have pretty much consistency within the positive regulators. But there is differential expression of different types of negative regulators if you're a deuterostome or a protostome, with some exceptions, of course, such as Strongylosis introitus, for example. And then CRY is broken into type 1 and type 2. Type 1 CRY, you'll see bind with or interact with the TIM per heterodimer, whereas CRY2 is really kind of acting in with the negative regulators. So really the question is, what's happening at the base of bilaterians? So for those that aren't really familiar with the terms, I had to give this to my animal science group because all they care about is mammals. You have your radial symmetry, which is going to be your Nematostella, and then bilaterians basically means you have bilateral symmetry. So what we're really talking about is, what does the ancestor look like right at this node where you have this gap between the anemones and the rest of the animal lineage? So how did the clock evolve from cnidarians to the arthropods or deuterostomes? And what does the mechanism look like in bilaterians? So using this species called Isodemetra pulchra, this is a worm, it's a marine worm that lives in between the sand grains. It feeds on diatoms. In the lab, it has been known to have a rhythm. Although it's anecdotal evidence, there's not really been any studies. This animal does lay its eggs at night. Ideally, there's a transcriptome available. There is a genome available. It's a little messy, and both of these are privately available at the moment. And these guys have been in culture for a while in Innsbruck, Austria, where they do a lot of this work, and we were able to do this in our lab as well. And so it makes it a great candidate to look at what happened with circadian clock evolution at the base of bilaterians in this group called the xenomorphins. So one question we really need to ask is, when did period appear? Nematostella doesn't have one. All the arthropods and deuterostomes do. So this could be a key point of circadian evolution. So we predict that this was actually going to be found at the base of the bilaterians and the Xenocoelomorphins. So if you look at the tree over here, you can see that, sorry, I need to move you guys. There is a Isodemetra pulchra period that roots with another Xenocoelomorphin called Haustinia, and it actually forms the basis of this period clade. And so the reason why we included a bunch of other orthologs is we want to make sure it's not a clock or something else, And so it's really kind of cool that it does form that base of that clade. And so when we look at the protein structure, we can actually see the Haustinia per and Nematostella per, Isodomitra per, both have 2 pass domains. That's indicative of per and mus purse for Drosophila and mus. And we can definitely see, again, there's nothing really going on with Nematostella. We can see that it does have a pass domain with aren't. All right. And then we also look to see what other negative regulators it has. It does not have a TIM1 ortholog, but it does have a Cry2 ortholog. And that Cry2 actually groups with the rest of the Cry types 2. And then down here, you can kind of see where the rest of the Cry1s that function differently in a different mechanism, for the protostome mechanism. Again, we have this photolyase domain, so it looks very much like a Cry. Per's there, sorry, my apologies. And so we can say that basically Isodomitra likely has a Cry ortholog. When we look at single cell RNA-seq, and this was done with a partner lab of ours who had already done this, the RNA-seq, we can see that there is differential expression between cell types, which is kind of interesting. And it's most highly expressed with both PER and Cry, are highly expressed in neurons, secretory cells, and digestive cells. However, this is a single data set. It's a single point in time. So we really don't know if there's variations across cell types. Now, when we take these proteins and we fold them and we look at interactions using Haddock predicting software, we actually show that Isodomitra per and Cry do form a bind together. They interact at this particular domain. This happens to be a part of the pass domain. We're currently expressing these proteins in E. coli and are trying to do some biochemical analysis to see if they actually will produce these heterodimers in vivo. So just to kind of summarize on this portion of the talk, PER likely evolved at the base of bilaterians, presumably in the xenocoelomorphins. We have verified this through sequencing that it is a real PER and a real Cry. And so what's interesting is we don't find a Cry type 1 or TIM1 in Isodomitra. So this kind of says that the PER Cry negative regulators are favored in the animal lineage. And so that means that the protostomes are probably more divergent. And so with that, we can kind of predict that we'd expect a PER Cry heterodimer that would negatively regulate the clock. However, much more work needs to be done to really show that in vitro or in vivo. So then we can ask the question, why did the clock evolve? Well, in previous research, specifically within Nematostella, there was a couple of different hypotheses, and there's been another hypothesis since then that kind of give a story of how the clock may have evolved. So we have endosymbiosis whenever you have 2 bacterial cells joining, you know, as a hypothesis as to why we may have a circadian clock. The circadian clock may have developed as a redox homeostasis mechanism first to mitigate free radical production. There's a hypothesis that suggests that it arose to minimize UV damage so that when you replicate your DNA to the dark phase versus a light phase. And finally, there is evidence to suggest that maybe there's a nutrient sensing mechanism that gave rise to the clock. So what is the evidence? And the question I really want to ask is, do these mechanisms predate the evolution of the clock, or does the clock come before the mechanisms? Just to kind of give us a little bit of an idea. So when we look at something like redox, we can see SIRT1. We know that SIRT1 does interact with the clock. It actually will interact with the EBOX and then have upregulation of PER and Cry genes. So there is an effect between the SIRT1 redox homeostasis and the interaction with the circadian clock. So when we look at Isodomitra in the xenocoelomorphins, we do see that it does have a SIRT1. And it does group with Nematostella SIRT1, and Neurospore has SIRT2. But basically what this is saying is SIRT1 probably came before the clock. Likewise, when we look at DNA damage response, we can look at the proteins of ATM and ATR. And so basically these are proteins that respond to single strand breaks or double strand breaks. And we have interactions with PER and ATM. We have interactions with Cry and TIM with ATR. And so they mitigate damage response to photoperiod. And so when we look at the proteins, we actually find that there is an Isodomitra protein of both of these, but there also are Nematostella proteins. So that indicates that, again, these proteins came before the clock. So we look at mTOR or TOR for nutrient sensing, kind of similar deal where we have, I pulled mTOR here, but Nematostella also has a TOR. So where we're kind of going with this is these mechanisms on these hypotheses may be somewhat accurate, but they only tell part of the story. So in conclusion, the Xenocoelomorpha is a keystone phylum that is really, really important in understanding the evolution of the clock mechanism because it fills this gap that we really don't have a lot of knowledge about. And so one of the key points is that per evolved at the basis of bilateral regions, which explains why you see per orthologs that are negative regulators in both the protostome and deuterostome lineages, but you don't see something in Nematostella. So Nematostella may have a different type of clock or may function quite differently. Again, these conserved domains and proteins within the redox and cell and metabolic mechanisms look like they predate the mechanisms of the circadian clock within the animal lineage. And so if we really still need to go back and verify that these Xenocoelomorphin sequences really are what we think they are. And so we need to do functional assays, pull them out, all that good stuff. So right now, this is largely just a bioinformatic survey, so to speak. But what it does is it gives us the ability to say that the proposed hypotheses are somewhat correct, but they don't fully explain the evolution of the clock. So in conclusion, we are taking all this information that we have and developing this new cohesive hypothesis that the clock evolved to coordinate homeostatic mechanisms within the light and dark cycle as the animals become more complex. And so with that, I would like to acknowledge a couple of people. So with Dr. Smith and Dr. Herbert at Winthrop University, have been instrumental in a lot of this work. Hannah Justin is a graduate student in North Carolina. He's helped with some of the bioinformatics. And then the single cell RNA-seq was actually done by Jules Giroux at the University of Freiburg with the same species. So we were able to kind of partner with him to get some of that single cell data. And with that, I'll take any questions.
Excellent, very cool talk. We have a hand up, so let me get that individual permission to unmute. Well,
can you hear me all right?
yes.
Oh,
Okay.
yeah,
Yeah, great talk. I just had a question because, I mean, you're talking about like these super far back, really conserved ideas. And I was wondering, and I mean, there's differences probably obviously in the mechanisms and the reasonings for why these come about, but is there, are some of these potentially even predating like the split between animals and plants as well? Like, because they both need
Right.
it, so.
Yeah, well, ironically, that's a really good question. Like Tor, for example, it looks like it may have evolved, over time with different lineages. So the Tor mechanism in plants does something different than the Tor mechanism does in animals, but yet they still use a very conserved protein that acts like a rheostat. Is kind of like almost like I want to use the term bottleneck, but with evolution, bottleneck means something very different than what I mean. It's as in like it's a rheostat in that one protein has a lot of different outputs. So there can be a lot of inputs to the Tor protein, and there's a lot of outputs to the Tor protein. And so like CERT and all that, they are highly conserved. And so that's why we kind of thought that maybe there's more a cohesive, way to explain this. And it's not any one mechanism. It's that as the animals, or as in this case, plants, because plants have the circadian clock too, but it's very different than animals, that maybe these mechanisms arose so that it can better coordinate all these other mechanisms within the light dark cycle. So for two reasons, maybe the mechanisms won't run into each other to cause damage because, every mechanism has byproducts that can be, negative in other mechanisms. And the other point would be that, by synchronizing with light dark cycle, it's also conservation of energy. And so those are two very vague hypotheses I can wave my hands about all day, but they're very difficult to actually show and prove because there's so many nuances and intricacies within all these different mechanisms that interplay with one another.
Yeah, it makes sense. Thank you.
Yeah, no worries. Any other questions? I think we have maybe 30 seconds for one more.
Yeah, Laura, you can put your slides up while we're still talking. I'm a little curious about, sort of the robustness to different, phylogenetic configurations, since, in that part of the tree, there's just a lot going on. How robust do you think conclusions are to kind of the changing landscape there?
Well, I mean, for the clock, it's pretty. I'm pretty confident in our data with the clock. Now, in terms of the other pathways, I mean, to be honest, like right now, our first question was which came first, the clock or this protein? And so, as you saw the trees, I probably can get better resolution on the trees with more species. But then, if you don't include the right species, then things can get more complex. Like if you add a nematode, for example, they're just kind of weird with some of their sequence structure, and it will throw off your entire tree. And so there could be that there's moments where you have animals that have diverged somewhat that make your tree a little bit muddy.
All right, thank you for that. All right, Lara, you ready to go? You are still muted. Let me make sure that you have the ability to unmute yourself.
Thank you.
There we go.
We're finding the unmute button. So I'm Lara Pereira. I'm a postdoctoral researcher in the University of Sheffield. And today I'm going to talk about lateral gene transfer in maize pangenome. So just a very brief introduction. Evolution is generally viewed as a vertical process, but the truth is that the branches of the tree of life can be pairs, and that means that the genetic material can be interchanged among different species. This is generally known as horizontal or lateral gene transfer, and it was initially very well known in prokaryotes, but more recently, over the last few decades, it was reported also in eukaryotes, in fungi, animals, and plants, although the mechanism of this happening is still unclear. At the end, lateral gene transfer just means to the gain of genetic material, but means different from reproduction. So essentially what we have is one gene that belongs to species 2 genome, somehow moves to the genome of species 1 that later has 2 copies of that gene, one that is the vertically inherited copy, and a second one that we call xenologue, or more generally just an ortholog gene that came from a different species. In our lab, for the last decade or so, my supervisors have been working in grass-to-grass gene transfer, so genes that are transferred from one grass species to another one. And recently, we identified LGT in 17 different grass species, and we saw that 13 of them have at least one lateral gene transfer in their genomes, which for us means that LGT is widespread in grasses. So it's a general process that happens in a high proportion of grasses. So these are the genomes that were used in that study, that were the ones that were available within the grass group at that point. In this case, we use only one reference genome per species, and we saw that the numbers were variable. And for example, in maize, that is, as you all, I'm sure, know, highly important grass species, mainly because of their importance as a crop from the Andropogony clade. We observed there 11 LGT. That was like a median number, so there's a few grass species that contain more, but within the important crops was quite a good number of LGT. So we decided to go further with maize. And one idea that we have, and we saw it in another species, is that pangenomes need to be used to quantify the full extent of lateral gene transfer in a species, because not all the accessions, all the individuals of the species will contain all the genes that have been transferred laterally. So we decided to use the maize pangenome, that is a very an excellent, very comprehensive resource that was published a couple of years ago to quantify LGT. So in this maize pangenome, 26 diverse accessions are included. There are these ones here. These same accessions and populations, genetic populations derived from them, have been extensively used for QTL studies and many genomic and genetic studies. And also transcriptomic data, methylation data were generated for the same data set. So it was like a very nice resource to try to investigate the lateral gene transfer landscape. We saw that in this paper that only about a third of maize genomes, maize genes are present in all the 26 genomes. And our question is mainly how many of them are LGT? Are LGT mostly core genes, so found in all the accessions, or only present in a few accessions? And does it mean that they are ancient LGT or more recent LGT? So to investigate this, we use the same phylogenomic pipeline that we have published before, or with very slight modifications. We started with the 26 maize assemblies, and we do a first blast filter. So we blast all CDS against all, and we select as candidates the genes for which the best hit is from a non-anthropogenic RAS. For those, we construct phylogenetic trees, and we use an automatic script, an automatic filter to select the ones in which the LGT candidate is nested in a non-anthropogenic clade. After that, we increase the sampling, so we use also transcriptomic data. We put a bootstrap filter, and we manually inspect the new trees to confirm that the LGT candidate is nested in a non-anthropogenic clade. And additional filtering, making sure that the tree makes sense according to the species relationships in general, that there are no paralogy problems, etc. And for those candidates, we do a topology test, and the ones that pass this test are our final LGT candidates. We end up with something like this. So here we have just one example of these trees in which we see a tree that makes sense according to the species relationships in general. We have a copy that is vertically inherited, so it's found in the place in the tree that we expected it. So here we have the vertical copy of this LGT, but then we have a second copy of these maize genes, an ortholog, that is nested within a different group, in this case the Centrini, and is in this case sister to the species Zuluaga bulbosa that would come back to it in a minute. So in the maize pangenome, we found 22 lateral gene transfers. It's only 0.02% of pangenes. So even though we observe LGT is a very rare process in grasses, in this case, in this pangenome, we saw that very likely all of them are old transfers, because in all cases we found non-domesticated relatives as Teosinte or Tripsacum, also nested with the LGT. And curiously, more than 50% of them were nested with Zuluaga bulbosa within the Centrini. So we would assume that this is the most likely donor, or a close relative to this species would be the most likely donor of this LGT. So here just a summary of the results. So these are the LGT candidates, and these are the maize accessions. And we just recorded the presence absence of the LGT in all the maize accessions and checked the copy number. And we can see that in some cases, in most of them actually, these lateral gene transfer are core genes, so they are present in all the accessions, all these ones in which we see a 1 here. But in some cases, they are variable. So we see that they are present only in a few accessions. So this first case, we have only 5 accessions containing the LGT. And we also see copy number variation, which is very interesting. For example, in this case here, we see from 0 to 7 copies of the lateral gene transfer, which could indicate possibly that is involved in local adaptation. And as we already said, most of them, the likely donor is within the Centrini group, and in many cases is sister to Zuluagaya bulbosa. Why is this important for us? Because Zuluagaya bulbosa is distributed in Mexico and the south of the United States. It's a common crop weed, and is the same region in which Sea maize was domesticated 9,000 years ago. So it makes sense biogeographically to think that Zuluagaya bulbosa could be the donor of this LGT because they were coexisting. To end up, we also looked at gene expression of this LGT in comparison with the orthologous native genes. And as you can see in the figure, if we see the general pattern, we see that the LGT is usually less expressed than the native gene in all tissues. But if we observe specific cases, there are specific genes that are highly expressed in certain accessions or certain tissues in comparison with the native gene. So this again could point out to local adaptation. So only certain accessions actually need that LGT, and therefore they are the ones that are duplicating and expressing at higher levels those genes. And just to end up, the take-home messages for me today are that LGT is quite widespread in grasses and can confer adaptive benefits. The number of LGT gained and retained varies among species and even among individuals within the same species. Therefore, a pangenomic framework is essential to characterize the full extent of LGT within a species. And in the case of maize, after analyzing its pangenome, we observed 22 LGTs, some of which are expressed and could have had a role in local adaptation. Thank you very much for listening. And these are the people in my lab that helped in this study. And yeah, I would like to acknowledge the funding. Thank you.
Excellent talk. And now we have time for questions. If anybody has a question, you can raise your hand and I can give you the power to unmute yourself. Otherwise, you are also welcome to put questions into the chat to be read off and answered by our speaker. Now you can kick us off if other people are feeling shy. So this is a fairly kind of complete study that you presented here. What do you intend to do with it next?
Well, I actually very recently got funding to sequence the Zuluagaya bulbosa genome. So yeah, very exciting. The idea is to sequence the Zuluagaya bulbosa genome and try to search for reciprocal transfers. So see if we also can find lateral gene transfer from maize to Zuluagaya, and also try to, if we have both genomes, we could compare these regions and see maybe how big these transfers are, because what happens is that in the context we are studying it, we are focusing only on genes, and intergenic regions are usually too divergent. But if we have the donor, it might be easier to also look at intergenic regions and see if we can find any kind of pattern of how big the regions are, how many genes are included. Can we see something on the junctions, so some kind of transposable elements or anything like that, that are facilitating somehow the integration of these genes.
Excellent. Congratulations on your funding.
Thank you.
I think we've got to start our changeover now. Our next talk is labeled as the last talk in our session. We will have one additional talk after that, someone who could not be accommodated in an earlier session. Addie, if you would like to put your slides up. Perfect, those look great. And you are welcome to start talking whenever you feel like it.
Okay, so hope you can hear me. So arguably the most well-known mutation in the world of biology is the human hemoglobin S mutation, which provides protection against malaria while causing sickle cell anemia in homozygotes. This mutation has served as a classic textbook example of adaptation by random mutation and natural selection. The story has been that one day it originated by chance in an individual in sub-Saharan Africa and then spread in the population there until its benefits were balanced out by its costs. But does this mutation really originate at random? So until recently, if one wanted to measure the rate of any particular mutation, that would have been impossible. Mutation rates have only been measured as averages across many genomic positions. But in our lab, we developed a method that for the first time has enough accuracy and yield to measure the de novo origination rates of particular mutations at particular base positions. And we used it to measure the rate of the HBS mutation in human sperm. Surprisingly, we found that it originates de novo more frequently in sub-Saharan Africans who have been experiencing intense malarial pressure for many generations compared to Northern Europeans who have not, and in the beta globin gene, where it provides protection against malaria, compared to the same mutation in the delta globin paralog, which does not provide protection. In other words, it originates de novo more frequently in the gene and in the population where it is needed. So the very first time the rate of a particular mutation is measured, it turns out this mutation is not random at all. This result counters the notion of random mutation on a fundamental level. So how could it be? So until now, there have been only two ways of thinking about mutation. One, that it's accidental, and another that it's Lamarckian, responding directly to the immediate environment. But I've proposed a different alternative called interaction-based evolution. According to it, mutations respond to biological information in a meaningful manner, and therefore they're not random. However, they do not respond directly to the immediate environment, and therefore they're not Lamarckian either. Instead, the rate of each particular mutation depends on information accumulated in the germline genome over the generations. At each generation, mutations are generated based on the information accumulated thus far, and those that survive become a part of the genetic information that will affect mutation origination in future generations. Therefore, evolution is like a long-term learning process based on non-accidental, non-Lamarckian mutation at its core. And this allows mutation to be directed in the long term. We argue that over the generations, mutational mechanisms gradually hone in on the genomic regions and on particular mutations in those regions that are most relevant for the currently evolving adaptations. Now, note that this does not mean at all that all heritable change is beneficial. Because this theory is not Lamarckian, it relies on selection to evaluate the consequences of mutation. And because it's difficult to change a complex whole, the mutational pressure can lead to genomic points of friction between the long-term process of adaptive evolution and the short-term consequences of recurrent genetic disease in accord with observations. But what is the mechanism that enables mutations to respond to information accumulated in the genome? A new study of ours begins to explore this question by looking at gene fusion mutations. As for all mutations, it's been thought that gene fusions occur by accident. It's been thought, for example, that the TRIM5 and cyclophilin A genes fused by accident in primates and by chance created a new gene protective against HIV-1. It's also been observed that genes that interact in one species are often found fused in others. But the belief in random mutation led people to explain that pattern too with accident. Perhaps random transcriptional read-through or deletion mutations fuse neighboring genes, and since neighboring genes often work together, this leads to a spurious correlation between gene interaction and fusion. Or perhaps many genes both near and far from each other fuse at random, but those that had interacted before fusion are more likely to succeed once randomly fused. But the theory of interaction-based evolution, which holds that mutations respond to internal information accumulated in the genome, led me to a different prediction, that repeated usage of genes together over the generations leads directly to their fusion via mutational mechanisms. Genes that are used together can become fused together. It is known today that when two genes work together, the DNA bends in 3D space to bring them with their chromatin open to the same place and time in the nucleus. Therefore, I predicted that by working together over the generations, genes would generate internal information in the genome about their joint action, which would increase their chance of fusion at their meeting place via any one of multiple downstream mechanisms, such as reverse transcription of the RNA, recombination-based mechanisms, transposable element-mediated translocation, and more. I also hypothesized that this mechanism would apply to both genes that serve somatic functions and genes that serve germline functions without invoking any Lamarckism, because the genetic information that indicates that two genes are working together, such as shared cis elements and transcription factors that bind to them, is available in the DNA and accessible in the germline for genes of all kinds due to the phenomenon of germline-specific transcriptional promiscuity. To distinguish this mechanistic prediction from accident, we analyzed several large publicly available data sets, including string and co-express-DB, as well as data from our own pipeline to look at interactions between genes in humans and gene fusions in other species and see what explains better the correlation between gene interaction and fusion. That it's due to accidental reasons, like random fusions between neighbors, or that it arises mechanistically from the interaction between the genes. We used several convergent measures of interaction, such as co-expression, co-localization of the gene products in the cell, the tendency of the two genes to be found in the same topologically associating domain, the semantic similarity of their associated Go terms, and result across all comparisons strongly supported the mechanistic prediction. Genes that interacted in humans were more often found fused in other species, whether they were neighboring genes or remote from each other, countering the hypothesis that random mutations fuse neighboring genes. Even among neighboring genes of the same intra-pair genomic distance, those that interacted more tightly were more likely to be found fused, which underscores the role of interaction in gene fusion. Also, genes more frequently observed in the same topologically associating domain in humans were more often found fused in other species, supporting our prediction on the nature of the mechanism. And the use-fuse effect applied regardless of whether genes served germline or somatic functions. We also looked at human cancer fusion mutations. Note that the selection pressures on a gene fusion to benefit organismal survival and on a gene fusion to drive cancer growth are very different from each other and can even be antagonistic. And therefore, from the perspective of random mutation, there shouldn't be any positive correlation between them. In contrast, the use-fuse hypothesis entails that mutational mechanisms are a primary factor limiting the set of gene pairs that can become fused, and therefore genes that interact are more likely to undergo fusion mutation both in evolution and in cancer, where the resulting fusion mutations could then undergo different selection pressures in each case, leaving a small overlap between evolutionary and cancer fusions. We found that indeed there was a small but statistically significant overlap between evolutionary and cancer fusions consistent with the use-fuse mutational mechanism. Now, we spoke previously about the fact that neighboring genes often interact, but notice that the use-fuse mechanism explains also why neighboring genes often work together in the first place, because the very same mutational mechanisms that fuse interacting genes can also often translocate them at first to the same neighborhood. And due to this and other empirical observations, we argue that the evolution of the genome architecture itself is largely driven by mutational mechanisms. This is in stark contrast to the view that the genome architecture is due to random genetic drift. So recall that the theory of interaction-based evolution argues that each specific mutation has its own probability, which depends on complex information accumulated in the genome over the generations. And this is demonstrated precisely by gene fusion mutations. There is no single fusion mutation rate. Each particular fusion mutation has its own probability, which depends on complex information accumulated in the genome, which involves all of the information that makes the two genes interact tightly, such as promoters, enhancers, transcription factors, and the epigenetic marks of the two loci and other loci that regulate them. These 2 points reverse the two key assumptions of modifier theory. But beyond the technical level, one of the most basic principles of learning and cognition by the brain is chunking, where pieces of information that are frequently used together become chunked or routinized through learning into a single piece that is thereafter activated as one. For example, when we first learn to drive a manual car, we must first coordinate the hand and leg movements to put the car in a particular gear, and with practice, the movements become fused into a unified sequence that follows automatically. It turns out that an analogous principle applies to molecular evolution. Separate genes that are at first activated by separate arms of regulation, after they're used together repeatedly over the generations, become fused together by mutational mechanisms into a single gene that is thereafter activated as one. So gene fusion mutations are non-random in a manner that makes them analogous to a key principle of cognition and learning by the brain. One might say that the HBS mutation is just one mutation and that the use-fuse mechanism is just one mechanism. But we argue that these principles generalize to mutations of all types, that mutations simplify and replace pre-existing system-level phenomena from which their origination follows mechanistically. We call this the replacement hypothesis. For example, we argue that gene duplication mutation is not random, but instead elevated transcription increases the probability of gene duplication mutation. And this further facilitates long-term evolution by alleviating the pressure on gene expression where high gene product quantities are needed, and by enabling specialization where multiple processes require the gene product and lead to high expression. In fact, we argue that the empirical nature of mutation embodies key principles of information acquisition. In our recent paper on the topic, we show how other mutation types and how the HBS mutation could abide by these principles. So where does all of this lead us? Darwin did not believe in random mutation. The notion of random mutation was proposed by Thomas Hunt Morgan in the beginning of the 20th century as a speculation. Darwin himself had a far more complex view of heritable change. In The Origin of Species, in the chapter devoted to the laws of variation, he wrote, "I've hitherto spoken as if the variations were due to chance. This, of course, is a wholly incorrect expression, but it serves to acknowledge plainly our ignorance of the cause of each particular variation." And in the conclusion of that book, he wrote that one day, a grand and almost untrodden field of inquiry will be opened on the causes and laws of variation. I believe that due to new empirical knowledge, new cutting-edge techniques, and new concepts, that day has come. I argue that there is no such thing as random mutation in evolution, but neither is mutation Lamarckian. Each mutation has its own probability of origination, which depends on complex information accumulated in the genome over the generations. When looked at in isolation, each mutation may seem to arise by a local genetic accident, but when understood in the proper long-term, large-scale context, mutations are a part of a vast system of interconnected mutational activity, an informational ecosystem of sorts. By replacing pre-existing interactions, mutations combine information and simplify it, generating from previous pieces of information new pieces that have the capacity to come together in emergent interactions at the system level. And this non-accidental yet non-Lamarckian nature of mutation enables long-term directionality in the origination of mutation, shapes the genome architecture, contributes to parallel evolution, explains the contribution of transposable elements to the evolution of gene regulatory networks, and more. Also, mutation origination rates at a high resolution present a whole new world awaiting to be discovered with empirical studies, and the principles of IBE are waiting to be explored with mathematical models and computer simulations. I would like to note the central contribution that Doctors Daniel Melamed and Evgeny Bolotin made to the research presented, and to thank my other lab members and our collaborators in Ghana and in Israel for all of their work, the John Templeton Foundation and the Israel Science Foundation for funding, and thank you all for listening.
All right, very nice talk. And just as a reminder, we do have one more talk after this that is not on the schedule. At this time, you are welcome to raise hands for me to unmute you, or you are welcome to put questions into the chat to be answered. All right, we've got a hand up from Alex. Let me ask you to unmute.
Hi, thank you. Thanks for a thought-provoking talk. I actually wanted to ask a question about the observation you started with, where you talked about an increased mutation rate at the hemoglobin locus in African populations. Is it that specific mutation, or is it a locus-wide increase in mutation rate? Right? Like, can you distinguish an increase in mutation rate for the adaptive mutation versus just a general increase in mutation rate?
Yeah, we actually see both things. So first, we see that the overall point mutation rate in the HBB gene in sub-Saharan Africans in the very specific six base positions we're looking at is overall higher in them than in Northern Europeans, which is already, you know, a result that counters the notion of random mutation, because this is a tiny 6 base pair region that has an enormous, plays an enormous role in adaptation, right? So if that overall point mutation rate in it is higher, that would not have been predicted from random mutation natural selection. It's in addition to that we see that in that 6 base pair region, the HBS mutation is the most is unique in that its rate deviates by far the most from its genome-wide average expected rate compared to all other mutations in that region, in that tiny region. So yes, you absolutely see a specific effect for the HBS mutation. In fact, in addition, you see that mutation rates vary on the single mutation level, which, you know, there's no population genetic theories really dealing with this, predicting this. There have never been any empirical studies looking at it because it wasn't possible. As I said in the beginning of my talk, all previous methods could only measure mutation rate averages. In population genetics, we usually talk about the mutation rate as though there's one such thing. Whereas I'm arguing here today that each mutation has its own probability. Not only that, but this probability is meaningful, and that the spectrum of mutation probabilities continually evolves and is central to the adaptations currently evolving, because you're looking at the long-term process. Also, let's not confuse it with the fact that mutation rates can vary between motifs. We're not just looking at motifs, we're actually looking at individual mutations in particular base positions, which has not been done before in any form or manner. I hope that makes sense.
Thanks.
It does. Thank you for sticking around to answer questions. We are going to move on to our final talk of this session, which is a bonus talk. Very exciting. All right, Berenice, you are welcome to start whenever you are ready.
Thank you. Voy a hablar en español. Espero que puedan activar los subtítulos en inglés. Puse las diapositivas en inglés, así les resulta un poquito más fácil. Buenas tardes. El título de la charla que les voy a brindar es Migración catádroma o adaptación local, el cangrejo nativo Cyrtograpsus angulatus en el río Chubut, Argentina, primer registro y análisis genético poblacional.
Un momento, Berenice.
Sí.
I just want to check that you have your, before we start, sorry, that you have your captions set to, so that you're speaking in Spanish.
Yeah, sorry.
No, it's okay. My translation was going a little weird, so. Si tu, I'm switching to French. En español, puedo configurar su configuración de subtítulos para
subtítulos en inglés.
Ya, si miras 3 puntos y más sur la abajo o en la parte inferior de tu pantalla de Zoom en la sección más.
Yes, it's okay now.
Yes, it's can you talk a bit more so I can check the translation?
Yes, my name is Berenice Troban. I'm going to talk today with you.
No, sorry, en español.
En español.
Sí.
Buenas tardes, mi nombre es Berenice. Les voy a brindar una charla titulada Migración catádroma o adaptación local. ¿Es okay?
I'm
¿Está
waiting to
bien?
see.
No, no.
Un momento, translation. See, yes, I would like help translating. My Spanish is muy mal. Juan, do you want to unmute? Or can we unmute Juan Rendon?
Yes, I've just given him unmute privileges.
Thank you.
Do you want I do something?
Hey there.
Oh,
I was just asking if you want to help with the translation. I believe I was talking with Katie, I think.
We just need to set up,
Karen.
yeah, we just need to set up the captions so that the translation on Berenice's side is in her language, speaking language is in Spanish. And so
Yeah.
that the captions don't go all weird.
Well, I mean, but I thought you wanted to explain to her how to activate the translation.
Yes. Because
and how do we do that? I'm sorry.
Okay, so on the bottom of the screen, there of your Zoom screen, there is 3 dots.
At the
And
bottom.
a more button, click on that. And then captions. And then set the speaking language to Spanish.
I'm sorry, what was that?
Set the speaking language to Spanish. So, and I don't know how it shows up in Spanish is my problem. So there's
Yeah.
the translate to, anyone can choose that for themselves, but on abajo, under speaking language, there's colons, there's English in my side. On Berenice's, on your side, it should be set to Spanish.
Yeah.
And sorry, it's
No.
complicado.
sí, sorry.
Oh, no, not your fault.
Berenice, ¿pudiste activarlo?
Yes, sí, y Juan,
Muy bien.
¿a ustedes les aparece bien? Yo puse traducción a inglés y el idioma hablado español.
The translations read all right now.
Yes?
Okay.
Okay.
Gracias.
¿Empiezo? ¿Sí? Sí.
Yes, go right ahead.
Bueno, buenas tardes, mi nombre es Berenice. El título de mi charla es Migración catádroma o adaptación local. El cangrejo nativo Cirtographus angulatus en el río Chubut, Argentina. Primer registro y análisis genético poblacional. Los decápodos evolucionaron en el Paleozoico como un grupo marino y desde entonces han tenido una radiación limitada en otros ambientes. Cerca del 80% de las especies existentes actualmente aún viven en el océano, en el mar. La colonización de ambientes no marinos requiere adaptaciones fisiológicas y las habilidades osmoregulatorias en general de los decápodos de agua dulce, más que nada de adultos y juveniles, son fuertes, sin embargo en los estadios larvales casi son inexistentes. Las migraciones diádromas en los estuarios son parte de una estrategia adaptativa que permite que el adulto viva en hábitats no marinos, como en los estuarios o en el agua dulce, en combinación con una conservación de un rasgo de vida ancestral, como la larva planctónica. Los colonizadores recientes generalmente muestran patrones intermedios como una fase de vida larval abreviada o desarrollo directo. Los decápodos tienen un ambiente ancestral marino y la conquista de otros ambientes es una adaptación posterior. Este cangrejo, la especie de Cyrtograpsus angulatus, esperen un segundito, ahí está. Habita zonas intermareales y submareales de las costas marinas rocosas, estuarios y bahías poco profundas. Tiene fecundación interna, desarrollo indirecto, larvas pelágicas de larga duración. Tiene una distribución amplia en Sudamérica, en el Atlántico sudoccidental. Fíjense que va desde el sur de Brasil hasta el sur de Argentina en Santa Cruz, y en el Pacífico desde el centro desde Isla San Lorenzo en Perú hasta Talcahuano en Chile. Y abarca un gran rango de salinidades y de temperatura. De salinidades está en el mar y también en los estuarios. Presenta esta migración diádroma que hablábamos antes, que quiere decir que el adulto está en el estuario o en el agua dulce y eclosiona la larva que, ayudada por la marea alta, migra hacia el mar donde se desarrolla, y la megalopa, el último estadio, es la que se vuelve bentónica y vuelve al estuario. Se convierte en juvenil y luego en adulto. Ese ciclo está presente en esta especie. Hubo un hallazgo en la localidad 28 de julio en el río Chubut. El río Chubut es un río que nace en la cordillera de los Andes, atraviesa la estepa árida y desemboca en el Océano Atlántico. A 112 kilómetros de su desembocadura hubo un hallazgo de esta especie cerca de un pequeño dique que se llama dique de Boca Toma. Esta especie, si ustedes miran en el río Chubut, la región estuarina está considerada hasta 4,5 kilómetros desde la desembocadura, es la línea azul, y la línea celeste es la influencia de las mareas, que llega hasta 10 kilómetros como máximo. Y esta especie está considerada marina y estuarial, pero se encontró bastante lejos de la zona estuarial. Se sabe, hay registros de los adultos en agua dulce, pero siempre muy cerquita al estuario, donde se reproducen, ¿no? Y también se conoce que el primer estadio del embrión no soporta salinidades de cero. Entonces nosotros, ante este hallazgo en el agua dulce tan adentro en el río, lo que nos planteamos fueron dos preguntas, dos hipótesis posibles, digamos. Primero, si existe una migración de los adultos a la zona estuarial a reproducirse, o si es que las hembras, luego de reproducirse en el río, migran a la zona estuarial para liberar a la larva y que ocurra toda esta migración. No es para, ¿no? Migran a la zona estuarial donde se libera la larva y migra hacia esta migración diádroma. Y la segunda pregunta que nos hacemos, si no es que existe esta migración, ¿es que hay una adaptación local al agua dulce? Entonces empezamos a buscar dentro de la familia de esta especie de cangrejo y encontramos que esta especie, que es el cangrejo chino de Manoplas, la traducción en español, presenta esta estrategia diádroma, pero además de esta estrategia migración diádroma, presenta una migración de los juveniles río arriba, y en el río pueden estar de 1 a 5 años, luego de lo cual los subadultos, antes de llegar a la madurez sexual, migran río abajo, y hasta pueden migrar 1500 kilómetros. Por lo tanto, lo que pensamos que puede ser que en nuestra especie ocurra esto, ya que no es, o sea, fíjense que si es 1500 kilómetros, nosotros lo encontramos a 112, es posible que ocurra esto en nuestras especies. Entonces el objetivo que planteamos fue primero reportar este cangrejo para el río Chubut, y luego estudiar la diversidad de la estructura genética a lo largo del río utilizando el citocromo oxidasa 1. La hipótesis que planteamos fue que la diferenciación genética de esta especie a lo largo del río refleja una migración de los adultos a la zona estuarial. Y la predicción es que los individuos a lo largo del río no van a mostrar diferencias genéticas. Entonces muestramos 3 puntos a lo largo del río, el primero en la desembocadura, el segundo en la mitad del río, acá está les muestro Trelew y 28 de julio. Mostramos 20 individuos por localidad, los frizamos, tomamos un pedacito de músculo y lo colocamos en alcohol 96 para luego realizar la extracción de ADN, la amplificación de la citocromo oxidasa 1 y el envío de las muestras para purificar y secuenciar en macrogen. Esos son los análisis que hicimos, el cálculo de los FCT, índices de diversidad, test de neutralidad y una red de haplotipos. Con respecto a los resultados, obtuvimos una matriz de 689 pares de bases, de 57 individuos, de 3 localidades, y en total obtuvimos 18 haplotipos, 15 sitios polimórficos, y de los cuales 8 eran informativos. Y con respecto a los FCT, si ustedes miran, lo que obtuvimos fueron valores significativos entre 28 de julio y Trelew y 28 de julio y Puerto Rawson, pudiendo separar estas localidades en dos poblaciones, una de Río, que es la más adentro, y las otras dos llamadas desde acá como estuarinas. Entre Trelew y Puerto Rawson no se observaron diferencias significativas, y estos son valores de FCT moderados y similares a los encontrados entre otras localidades mucho más alejadas, entre 500 y 1800 kilómetros de distancia, en un estudio que todavía no fue publicado de esta especie, a lo largo de su distribución. Por lo tanto, con respecto a la hipótesis, si la diferenciación genética refleja esa migración catádroma hacia el estuario para reproducirse, no habría soporte de esta hipótesis, tendría que ser rechazada. Luego calculamos los índices de diversidad entre estas dos poblaciones y lo que pudimos observar a grandes rasgos es que la población del río mostró mayor diversidad, tanto diversidad nucleotídica, haplotídica, como el número medio de diferencias pareadas. Con respecto a los test de neutralidad, vimos con el test más sensible, que es el FDFU, que ambas poblaciones mostraban una expansión poblacional reciente o evidencias de selección. Con un solo gen no podemos diferenciar cuál fue la causa. Y cuando vemos la red de haplotipos en estas dos poblaciones, vemos que, por ejemplo, el haplotipo 1, el 3 y el 4 están compartidos entre ambas poblaciones, y luego hay el 80% del resto de los haplotipos estuvieron representados una única vez, o están en una población o en la otra. Con respecto a la discusión, entonces, la diferenciación genética encontrada nos hace pensar que es el resultado de probablemente que la población del río, sin pensar en una migración, sin pensar en una adaptación local, es que se esté reproduciendo en el estuario también, que ocurra una migración, pero que el tiempo de reproducción sea diferente que el de la población estuarial. Y luego podemos pensar en la adaptación local, que podría ser un poquito menos probable, pero podría ocurrir que entonces se esté reproduciendo en el río, la población llamada río, ¿no? Y la del estuario en el estuario. Entonces puede haber un aislamiento temporal o aislamiento espacial que podría estar reduciendo el flujo génico entre estas poblaciones. Y pensamos que con datos de la biología reproductiva se pueden poner a prueba. Con respecto a la reducción de la diversidad genética, que el río tenga mayor diversidad que la población estuarina, pensamos o lo asociamos a la presencia de esta especie invasora de 2015 en la región, que no solo es marina, sino también es estuarial, y que está desplazando, se ha visto, a todas las especies en la costa, a todas las especies nativas, y a esta también, a Cyrtodapsus angulatus. Entonces puede ser una de las causas. Con respecto a los test de neutralidad, como yo les decía, con un solo gen, ustedes deben saber mucho más, porque estamos en este módulo que saben más de este tema, que necesitamos otro gen para saber si se debe a un proceso demográfico o a una evidencia de selección. Y algunas observaciones interesantes que hicimos con los datos que tenemos por ahora es que, por ejemplo, la población de río, los ejemplares son mucho más grandes, el caparazón es mucho más grande que los ejemplares de la población estuarial, y se ve en este gráfico, ¿no? Con respecto también a los datos que tenemos en algunos meses del año y a lo largo del río, vemos que hay presencia esto no está en inglés, hembras y machos en diferente tiempo a lo largo del río, por lo cual podría estar indicando de alguna forma una migración o menor actividad de las hembras o de los machos en cierto momento del año. Y también hicimos algún muestreo de biología del desarrollo gonadal, y lo que hemos visto que la población del río, los ejemplares no tienen la gónada desarrollada, a pesar de tener un tamaño muy grande. Entonces eso nos llama bastante la atención y nos hace acordar al caso de la otra especie dentro de la familia. Entonces, bueno, lo que resta y lo que vamos a empezar en julio de este año es hacer un muestreo más durante un año o dos, dependiendo de los resultados, incluyendo 5 localidades, una inclusive fuera del río, y hacer un estudio de desarrollo gonadal, de abundancia, del radio de sexos, para poder poner a prueba las hipótesis que les planteé anteriormente. Con respecto a las implicaciones de conservación que tiene este estudio, es importante conocer, ya que en esta especie está esta migración diádroma en los estuarios y un desarrollo larval extendido que habla de conectividad entre las poblaciones, pero en este trabajo que les decía que todavía no está publicado de la especie, habla de una alta diferenciación, alta divergencia entre las poblaciones estuariales, que hay poco flujo, a pesar de pensar que si uno tiene un periodo de larva extendido, la conectividad va a ser mayor y va a haber menos diferenciación. En este caso no ocurre, y vemos que dentro del río la diferenciación encontrada es similar a otras a lo largo de la distribución con mucha más distancia geográfica, ¿no? Entonces, es importante este conocimiento para la conservación de esta especie. Y además, si existiera esta migración, también es importante la planificación de construcciones a lo largo de los ríos, no solamente el río Chubut, porque probablemente si es algo característico de esta especie y todavía no se conoce, no pase solamente en el río Chubut, sino a lo largo de toda la distribución en muchos otros ríos. Entonces la construcción de diques va a afectar bastante a la biología de esta especie. Y también teniendo en cuenta que hay una especie invasora que desplaza a todas las nativas, incluso a Ungulatus, entonces conocer la genética poblacional y cuántas poblaciones hay de esta especie y con cuánta divergencia es importante. Así que muchas gracias, y si pueden comentar, ya sé que no están relacionados a este tema, al de la sesión, pero cualquier sugerencia o comentario va a ser más que bienvenido. Muchas gracias.